Well, hello there. Welcome to topic 2.5, enzymes. All right, let me make this bigger. In our last couple days, last couple lectures, we've been talking all about proteins, right? The building blocks of proteins, how amino acids are made, peptide bonds between those amino acids. And as those amino acids build up, they make proteins, right? The importance of proteins, that proteins do everything in our body, right? That they have genes have all the directions, but what they have the directions for is making the proteins. And one of the most important types of proteins in our body are enzymes. Right? And this is what we have our lab on. And we're going to look at enzyme reactions, the different types of enzymes, and all why it's so important for them to keep their shape. All right. So enzymes, types of proteins. Let's jump right into it. Okay. But at the end of this lecture, again, you don't have to write this down, but we will be going over, these are kind of like that quiz that you might want to take at the end. What are substrates? What is a catalyst? What are the different interactions? What makes something specific? My particle theory collision, factors, and then some applicable real life, or not real life, everything's real life, but some more common examples of enzymes and what we're doing to try and alter for people that are lactose intolerant. Okay. All right. Let's start out with just what is an enzyme. Okay. So an enzyme, you can just straight definition is anything, a protein in your body that acts as a catalyst. And a catalyst, the definition of a catalyst is going to increase the rate of reaction. It starts chemical reactions, right? So you might have used the word catalyst like in English, like this served as a catalyst for her, moving on to college, or it's like something that starts an action, right? So biological catalysts are going to increase our rate of reaction. They help the reactions start and then happen quicker. Okay. Now you need to know like the basic kind of vocab for this. So if this is my enzyme here, this little Pac-Man, this is my protein, right? The substrate, right, is the molecule that an enzyme reacts with, right? So to form this product. So I have the enzyme, I have the substrate. If you can kind of see right here, this substrate has a very specific shape. The enzyme has a specific shape. And this binding site right here, this is my binding site, together makes the enzyme substrate complex. Okay. Now you can think of this substrate as like the thing that is going to be broken down. It's the like, whether it's a molecule or another type of protein, right, is that this thing, substrate, binds to the enzyme, and then I get these products. So they come together, the enzyme breaks them apart, and the enzyme is not used in the reaction. It's not altered, right? So only the product is. All right. This place where, make sure that you have that down, this place right here where the two things bind, that is called the active site. Here we go. Sorry. Let me end the show here. Something like that. Go to my pen, right? So right here, this is called the active site, right? This is called the active site, right? And then make sure we know that my enzymes they're not used in that reaction. Okay. All right. So what do we use? There we go. Or what does a catalyst even look like? Or what does it mean? Some analogies for what that would look like with frogs. Analogies with frogs. There's a couple different visuals. I took up like this lecture, and then I have your book open as well. So hopefully I can give you a couple different ways to look at this, right? But here's an analogy for the catalyst. So the reason why it speeds up those chemical reactions is because it lowers the activation energy threshold. So the activation energy is the amount of energy that's needed for a reaction to start, right? So it's like, as soon as I have this much energy, then the reaction can actually move forward. What an enzyme does, hold on, I'm getting a phone call. Oh, no, I'm not anymore. Let's see if they call back. What an enzyme does is lowers that amount of energy that's needed. So instead of like having to have this much energy, it lowers it. So it actually reduces the amount of energy. It's an efficient way to break down that substrate by lowering the activation energy. Okay, you don't need to draw these frogs here, but it's probably good to know that's what we're looking at. Let me show you really quick a different graph. Okay, found it. So here's a more scientific, mathematical way to look at lowering the activation energy that does not have frogs, right? So here's something you'll see in your book. You've probably seen this before in biology, right? But if I'm looking at here, my energy, right, required on my y-axis here, and the progress of the reaction, so the reaction going through. So here is this, like the amount of energy that's needed without, right? So without the enzyme, right here, still goes up to the same products, right? So the reactants and the products are the same, but here's the course of the reaction with the enzyme, right? So the enzyme goes here, it takes less energy. So you're basically, it's a more efficient way to break down, right, for your body to break down substrates. Here's another couple, what was I going to say? I liked this one too, right? A visual for that as well. So I've got the substrates here. I've got the active site. They come together, they make the complex, right? And they break those things apart into a smaller unit. But again, with the enzyme not being changed within that reaction. Okay. All right. So there's two different ways, right? So you need to know the two different ways that that enzyme and the substrate like come together, the two different types of interaction. One is called the lock and key. I love when things are just simple and kind of tell exactly what's happening. And the other one is the induced fit model. Okay, so lock and key and the induced fit model. So right here we have the lock and key, and this is kind of what we just talked about in that last example. I have a very specific site, right? Or I'm sorry, very specific shape on my enzyme. I have a very specific shape for the substrate, and they fit together, right? And we call this concept here specificity, right? Specificity, right? This explains why those two things fit together. Now, the induced fit model, right, to induce, right, or to change or to start, right? This active site, if you look right here, it's not rigid, right? It's kind of flexible. And so when it binds with the substrate, it can actually change to make that substrate or like to fit to the substrate, right? So the lock and key, this is completely stuck in shape. It does not fit. It's looking for that specific substrate. But you can see right here by the induced fit model that because it can conform and change, it can actually work with a couple different types of shapes there. Okay, so two different interactions, lock and key, and then that induced fit model. All right, so going back to the specificity, I do not like that word. Say that out loud 3 times fast. This active site right here, because it has that very specific shape, right? If it is damaged in any way or denatured, what we'll look about in our lab, it's going to either change the enzyme reaction or it's not going to happen at all, right? So there's a couple different ways that we can change this site. But the specificity, right? If I look at like how this shape went to this shape, now it's not going to break down that specific substrate anymore. Okay, so this slide says that enzyme activity is highly dependent on the tertiary structure, right? The tertiary structure. That's looking at how things are built, right? We looked at like the collagen, right? How that's come together. Even the, the hemoglobin that we built in class, right? Has a very specific shape. So if I change the active site, it's going to change. It's either going to completely disrupt or diminish enzyme activity, right? Those two things. Oh my gosh, hold on a second. Okay, apologies. So there's a couple different ways that proteins are denatured, and that's what we'll be looking at in our lab, and we'll be looking about in our body, right? So why things can be so harmful for us sometimes is because those proteins, the enzymes, are denatured and change their shape. Okay, there's also good reasons, right? Or when we look at like denaturing occurring, so I'm thinking, who was it, Jen? In third period, we're talking about baking, right? Changing the shape when you're looking at cooking eggs. There's a lot of ways that when we denature, it's a good thing. But in our bodies, we do not want that to happen often because it changes like the actual active, what the heck, active site. Oh, that's okay. Okay. So another theory, right? Particle collision theory. So how does that work? So you can think about like, I've got these little substrates that are, bouncing around. I have my enzymes, right? Without a specific type of, when I say like magnet or force coming together, the particle collision theory explains why those, or how those two things are going to come together. All right. So if you can go back in your memory to thinking about like liquids and like how particles are broken up, and we even talked about in water, right? What we know about liquids is that they're always colliding, right? They slide past each other. And so for those two enzymes to interact, the particle theory says that if I'm going to increase the enzyme activity, I have to increase the amount of collisions that are occurring, right? So if I want my enzyme activity to increase, if I want the substrate to be broken down faster, I need to change the rate at which the things are bumping into each other so that it's more likely that they will bump into each other. Okay. Now there's a couple ways we can do that. One, by increasing the level of the motion, so thermal energy, right? So we know that as I increase the heat, it's going to increase the movement. That's going back to physics and probably your chemistry, and that's going to increase the particles coming in together. Okay. So if I look at this visual here, okay, just kidding. I was right here. If I look at, oh no, go away. If I look at this one right here, oh my gosh, stop, cancel. Oh, no, I don't want to restart. Cancel. Oh my gosh, you guys, this is driving me crazy. Here we go, 2.5. This is in your book. Another kind of visual way to look at how like that substrate, these water molecules are bouncing around, the substrates are bouncing around. Here's my active site. So if I increase the rate at which those, this is sign language, right? At which those water molecules are moving around, I'm going to increase the probability that those two things are going to come together. Okay. All right. So now let's look at some factors that change or increase or decrease enzyme activity. Okay. So because that enzyme is not consumed, right? It just stays there. It's not, that enzyme doesn't change. We can actually reuse them over and over and over. And so if I have the enzyme at low concentration in the body, if I have a high concentration of the substrate, that enzyme is not used up. So it's going to keep reacting and reacting and reacting. Okay. So what that allows us to think about, like the efficiency of that in the body. If I can reuse that enzyme over and over and over, I don't have to have as many, right, produced, right? So you're saving energy, it's efficient, and I only have specific enzymes in low concentrations because they can be used over and over. Okay. Now there's two different factors, or actually there's a couple different ones that we're going to be looking at enzyme activity right here, right? So the factors that influence the frequency of the collision, right? So how fast or how often those two things are colliding. And then how, like, the capacity for the two to interact, right? So looking at if things are altered or changed in shape, what are some things that affect those? Okay. The two things we're going to look at in your lab are temperature and pH, but then another factor that would definitely check or affect how fast this enzyme is going to act is my substrate concentration. So substrate is the thing that's being broken down, and concentration is the amount in the water, right? So if I've got high concentration or low concentration, changing the rate at which those two things come together and react. Okay. All right. So the temperature, right? So looking at the temperature is one of those factors. So lowering the temperature, think about what that's doing to the particles that are bouncing around. So if I have less movement by the molecules, that lower temperature might not have enough thermal energy for that reaction to occur, right? So when I lower the temperature of the reaction, just like in the general, like I get less particles coming in together. Now, if I increase the kinetic energy, I'm going to increase the amount of collisions. So specific enzymes and places in your body will have optimal temperature where I reach that peak, right? Where I want the perfect amount of where it's moving around a lot, but not changing the shape of the enzyme. Okay. Because if I change the temperature too much, like when you boil or cook, right, it's going to actually completely change the shape. It denatures, it breaks down the protein. So I want to increase it to like the specific place where I get the most rate of reaction and I'm not denaturing the protein. Okay, so that's one of the factors. Another one is pH, right? So looking at acids and bases, there are enzymes that act at certain places where their pH value is what we call optimal. All right, so if I change the pH, it's going to change the charge and then the solubility and all that other chemical stuff we've talked about, right? And so enzymes in your body actually have really specific pHs where they're at their optimal range. So there's a wonderful, Smire's just popping around, of a stomach right here, right? So this is looking at all the different enzymes in your body. Think about like your stomach and how acidic that is. So this is the little enzyme, obviously, this little guy breaking things down. Enzymes like pepsin and lipase, these are enzymes that live in the stomach and exist in the stomach, and they function at a very low pH, right? Because I've got more acids. Now, in your pancreas and your small intestine, I have different types of enzymes because they function at a different level, right? So enzymes in my blood that are in a pH of, of water, about 7, they're not going to work as well. They might be denatured in the stomach, right? And then the opposite way around. These enzymes will probably not function well in a higher or basic environment. So we have different enzymes in different places in our body because there's different pHs in our body. And those enzymes help break that food down, help it absorb, get our nutrients, all that fun stuff. Substrate concentration, right? So substrate concentration is the amount of the thing that's being broken down, right? And so because my enzymes stay the same, right? So you imagine like I have these enzymes and they're staying the same. If I have low concentration, right, that rate of reaction, as I increase the amount, so I only have a few, the reaction's slow. But as I get to like here, so I'm getting more and more in the blood, it's going to increase the amount of interactions, but then it hits this plateau right here. This is where I've used all these enzymes and they're being used over and over and over. And so they can't go any faster because the enzyme amount is limited, right? The enzyme activity plateaus, levels off because that saturation, and there's only so many places that the enzymes can attach to the solute. Okay, here's a couple examples, some things we've actually already talked about. And that I'll have you guys copy down in your activities. So you do not need to put this into your notes. But here are some things that we can actually see. We can do different, experiments with. For instance, we are using catalase as our enzyme in our lab, and we're breaking down hydrogen peroxide here. And the product that is broken down, so it's released, is oxygen. Okay, so I'm getting that oxygen gas. We are going to be looking at bubbles or the amount of production, but you can also look at starch being broken down by diastase. You can look at different proteins with pepsin being broken down, and we can actually see a color change because the protein goes away. So there's a lot of different ways that we can see enzyme activity in labs, and we will need to know some of these certain things, and I'll talk about that in class. Okay. All right. So talking about lactose. So how do I actually, so the people that are lactose intolerant, right? In society, there's it in here? It's not. Okay. Lactose is a protein, and some people do not have the enzyme that breaks down lactose. Okay. And so these enzymes, like when they fix to a static surface to improve their, when I can actually stick them to a surface to improve their reaction rate, sometimes we get this increased yield of breaking it down. Okay, so this is milk with lactose, and then these are my lactose beads. I'm sorry, we're talking about how to get rid of the lactose in there. And by putting those lactose beads together, I can actually this is talking about creating lactose-free milk. I can actually put that in there, and it actually takes out the lactose, breaks it down, and I get this lactose-free milk. This whole process is used by using immobilized enzymes, making them stay in one place, breaking them all down very easily, and because they're all concentrated, and then creating something new. Okay, so lactase, it's purified by yeast or bacteria bound to this substance, and then milk is passed through. This is in your book. Hold on, right here. There's a cool thing in your book about looking at the different populations of lactose intolerant individuals. Ooh, it's got to be way down here, right? Because that would be medical. Oh, here it is. Okay, so looking at populations of people that are lactose intolerant. So we can tell, like, obviously genetic, right? Because our genes are creating that enzyme that breaks down lactose. That in certain parts of the world we get, probably because of similar DNA, we get higher populations that are lactose intolerant. Okay. Now, lactose, another cool thing that they're doing that they're playing around with is actually producing cows that produce lactose-free milk, right? So there's where would I looking at that? That's in the other one. So I'll show you that in the other one. But this has a lot of different useful applications, right? We've got the dairy-free for people that are intolerant. We can actually increase sweetness instead of using sweeteners, and change where ice cream crystallizes. So getting down that temperature, and then producing various cheeses and yogurts in a timely manner. Okay. Now, in your book, hold on. Oh, boo, I can't find it, so I'm not going to waste time there. But they're, instead of trying to create lactose-free milk by taking it out using the yeast, they're actually they're playing around with creating cows, right? Genetically creating cows that create lactose-free milk, which I think is pretty darn cool. Okay. One other kind of visual here, looking at what enzymes do, are used and usually mobilized. So those different places that we use them. And then we'll look at this a little bit more in class, like looking at how lactose, right? So breaking that down. They also have, you probably just stopped listening, but formulas for babies that protein's too big for their stomachs to be broken down. They also have some really expensive formulas you can buy with it's already broken down for you. Okay. All right. I will give you this kind of summary thing in class, but that is our lecture on enzymes. Lots of stuff to digest, to break down, and we'll do some more practice with that in class. Have a fabulous day.